MIT researchers have developed a machine learning based technique to more quickly calculate the binding affinity of a drug molecule with a target protein. Drugs can only work if they stick to their target proteins in the body. Assessing that stickiness is a key hurdle in the drug discovery and screening process. New research combining chemistry and machine learning could lower that hurdle. The new technique dubbed DeepBar quickly calculates the binding affinities between drug candidates and their targets. The approach yields precise calculations in a fraction of the time compared to previous state-of-the-art methods. The researchers say DeepBar could one day quicken the pace of drug discovery and protein engineering. And they say their method is orders of magnitude faster than before, meaning we can have drug discovery that is both efficient and reliable. The research appears in the Journal of Physical Chemistry Letters. The affinity between a drug molecule and a target protein is measured by a quantity called the binding free energy. The smaller the number, the stickier the bind. A lower binding free energy means the drug can better compete against other molecules, meaning it can more effectively disrupt the protein's normal function. Calculating the binding free energy of a drug candidate provides an indicator of a drug's potential effectiveness, but it is a difficult quantity to nail down. Methods for computing binding free energy fall into two broad categories, each with its own drawbacks. One category calculates the quantity exactly, eating up significant time and computer resources. The second category is less computationally expensive, but it yields only an approximation of the binding free energy. The MIT researchers devised an approach to get the best of both worlds. DeepBar computes binding free energy exactly, but it requires just a fraction of the calculations demanded by previous methods. The new technique combines traditional chemistry calculations with recent advances in machine learning. The bar in DeepBar stands for Bennett acceptance ratio, a decades-old algorithm used in exact calculations of binding free energy. Using the Bennett acceptance ratio typically requires a knowledge of two endpoint states, example, a drug molecule bound to a protein and a drug molecule completely dissociated from a protein, plus knowledge of many intermediate states, example, varying levels of partial binding, all of which bog down calculation speed. DeepBar slashes those in-between states by deploying the Bennett acceptance ratio in machine learning frameworks called deep generative models. These models create a reference state for each endpoint, the bound state and the unbound state. These two reference states are similar enough that the Bennett acceptance ratio can be used directly without all the costly intermediate steps. In using deep generative models, the researchers were borrowing from the field of computer vision. It is basically the same model that people use to do computer image synthesis. While adopting a computer vision approach to chemistry was DeepBar's key innovation, the crossover also raised some challenges. In tests using small protein-like molecules, DeepBar calculated binding free energy nearly 50 times faster than previous methods. DeepBar has the exact same accuracy as the gold standard, but it is much faster. In addition to drug screening, DeepBar could aid protein design and engineering, since the method could be used to model interactions between multiple proteins. In the future, the researchers plan to improve DeepBar's ability to run calculations for large proteins, a task made feasible by recent advances in computer science. This research is an example of combining traditional computational chemistry methods developed over decades with the latest developments in machine learning.